All right, we're going to go ahead and get started with our next presentation here. This one is sponsored by Enzymotec, and we're going to be talking about the bioavailability of krill oil, results of a new clinical study. Again, this category of omega-3s, DHA, and EPA, a hot one, and krill oil, one of those hot topics within it. Our speaker, Dr. Sigalit Zahut, Manager of R&D, Division of BAI at Enzymotec, will be presenting. And she joined Enzymotec in 2008, working as the company's regulatory affairs manager. In 2010, she was appointed to manage the R&D team. And in this capacity, she's responsible for Enzymotec's division of bioactive ingredients, that's the BAI, overseeing clinical studies and managing the intellectual property and regulatory affairs. In addition to a Bachelor's of Science degree in Life Sciences and a Master's in Biochemistry, she received her PhD from Weizmann Institute of Science in Israel. So, again, our company is all over this omega-3 issue. Krill oil is a hot topic, so we're excited to have you here to talk to us about this and the results of the new clinical study.
Thank you. Okay, so hi, everybody, and thank you for being here and listening. So as I was introduced, I'm going to talk to you today about the bioavailability of krill oil, and I'm going to present for the first time in public the results of a new clinical study. I will talk a little bit about Enzymotec, about krill and krill oil, about omega-3 bioavailability, and in length about the clinical study, the results, the protocol, and a little bit of discussion. So Enzymotec is a global developer, producer, and supplier of innovative lipid-based bioactive solutions for the healthcare and nutrition markets. We are privately owned. We are located in the northern part of Israel. As you can see here, this is our manufacturing plant in the northern part of the country, and we also have joint ventures with other countries such as India and Sweden. We have 3 independent divisions. One of them is called Biopharma, dealing with medical foods. We have the infant nutrition division, obviously dealing with infant nutrition and developing a special component of fat mimicking mother's milk, which is put into infant formulas. And the bioactive ingredient division, the division that I am part of. I'm the manager of the R&D for this division. Our division deals with nutrition, with dietary supplement, as well as functional food. Our company was always based on lipids. Lipids are not very simple molecules. They require a lot of very sophisticated technology, and we have made it our business to deal with lipids for the past decade. A lot of our products are based on phospholipids and omega-3. Our company offers comprehensive services and solutions. We have the R&D, which I manage, and we do intellectual property, we run clinical studies, we do regulatory affairs. We also have a process development team with very capable engineers that work on innovation of lipid products and offer a variety of applications and support to our clients. We also have a manufacturing plant where we produce our ingredients. And we also have, of course, the marketing, which deals with, selling our products. The bioactive division mission plan. We have 2 categories of products, of ingredients. One is in the cognitive solutions. We have a variety of phosphatidylserine ingredients and GPC. And in the general health solution, we have the krill and another product called omega-choline. So what is krill and what is krill oil? Krill, and this is a swarm of Antarctic krill residing in the Southern Ocean, is a pelagic crustacean. You can see the red color. It's very typical, and it comes from the astaxanthin. This Antarctic krill is the source, the organism used to produce krill oil. And what is krill oil? So krill oil, unlike fish oil, is comprised of phospholipids, of triglycerides, and as I said, astaxanthin, which is a very powerful natural antioxidant. The phospholipids are bound to both EPA and DHA, as well as the triglycerides. Phospholipids are very different from triglycerides. While triglycerides are a fat used for storage of energy in our body, phospholipids are actually biologically functional molecules. And every cell in our body is surrounded by the plasma membrane, which is comprised of phospholipids. Our brain is primarily made of phospholipids. The production of krill oil is very different from the production of fish oil. While the fish biomass is being cooked and pressed and then refined using deodorization in very high temperature to yield triglyceride-based oil, the krill biomass is again cooked and pressed right on the boat immediately after harvesting to avoid spoilage, but then the extraction of the oil is done in a very different and more mild way compared to the fish oil. So actually, the extraction of the oil from the krill produces crude krill oil, which is not a triglyceride-based fat. We in Enzymotec add another step to the crude krill oil, which we call the MSO, the multiple step oil extraction. And by using this method, we eliminate the use of aggressive chemicals and high temperature typical to refining process, and we use special solvents and controlled steps. With the MSO, we have a special type of extraction technique modified exclusively for marine and krill biomass, so the oxidizing and destabilizing impurities are removed. And an example for that are the total volatile nitrogen. These are compounds that we know from the smell of rotten fish. For example, trimethylamine is the type of smell that you have when you have decomposition of fish, the smell of nitrogen compound. And we manage with the MSO to actually get rid of all those compounds, and we even have it on our certificate of analysis to make sure that the levels of TMA and TVN are very low and almost are undetectable. Here we can also see that the MSO keeps the level here of those compounds, the TVN, almost undetectable, where if you look at other krill oil, you can see that with time you have a decomposition which yields higher levels of TVN. Similarly, by removing all kind of destabilizing impurities from the krill oil, we manage to keep the azoxanthin to be very stable and at very high level at all times, while in other krill oil, it starts to decompose and to degrade very rapidly. Krill oil was a subject of many studies already from 2003 that looked at different indications and health benefits, from inflammation to management of hyperlipidemia, and a lot of talk about bioavailability. Actually, krill oil was shown or displayed as having higher bioavailability compared to fish oil. And there were a couple of studies done with krill oil. So is bioavailability of omega-3 bound to phospholipid actually better? Well, the idea that phospholipids are more efficient carriers of omega-3 into the blood and target organs was not new. And multiple studies have shown that with animals, that when you feed them with phospholipid-bound omega-3, you actually have better absorption of DHA or EPA in the target organs, the heart, the brain, and so on. However, when it comes to krill oil, there are a couple of publications a few years ago when they're trying to compare fish and krill oil, but actually failed to show any real statistically significant improvement in the bioavailability of omega-3 from krill over fish. Another publication that came quite recently actually is looking at kinetics at very short time with acute dose of omega-3 and then following up over 72 hours. And they could report that there is a trend of better EPA absorption, but that's it. So based on the above, krill oil was suggested as a better omega-3 carrier, but there are no studies to actually show that this is true and to prove it in a statistically significant way. So that was one of the first assignments I got when I became the manager of R&D. And we had to really do a lot of research and come up with a good plan of how we're going to prove something, which obviously others that are very good and capable tried and failed. So the study objectives was to demonstrate that omega-3 bound phospholipids derived from krill oil have superior absorption compared to fish and to placebo. And this is following 4 weeks of supplementation. Absorption was measured by comparing levels of omega-3 fatty acids in both the plasma and the red blood cells. Now, the red blood cells are very important to test, and I will discuss it more in length later. But this was also never shown with krill oil before. So the study design. We did a study in the University of Manitoba in Canada. Professor Peter Jones, who's the head of the Richardson Center for Functional Foods and Nutraceuticals and ran a clinical study center, was the principal investigator. The population of the study, we had 24 healthy subjects. However, because the design was a crossover design, the same 24 people were actually treated with all 3 interventions, the fish oil, the krill oil, and the placebo. We treated the people, those healthy people, with 600 milligrams of omega-3. We made sure that we have the same exact amount of omega-3, total omega-3, in both the krill oil and the fish oil, so we can do a real comparison. And the exploratory and the endpoints were omega-3 fatty acids, as I said, in the blood, both in the red blood cells and the plasma. So this is just to show you what the design looks like schematically. So a crossover design is where you start 4 weeks of treatment and then you have to have a washout. So the system, the blood is emptied from all the intervention, from the krill and the fish oil and the omega-3. And then you have another 4 weeks of treatment, and then you have another washout, and then the final treatment. And of course, the people are randomized, so each are getting their intervention in a different way. This design is very good to eliminate noise that come from the difference in metabolism that people have from one another. On the other hand, it is not easy to manage a very lengthy study where you need to keep the people on a certain regimen for such a long time. This is to show you the treatments. So as I said, we have compared the total omega-3. But because the composition of krill oil and fish oil is so different, it was impossible to actually have it exactly the same. But we did try to get as close as we can with the DHA and the EPA. This is just briefly to show you the inclusion criteria. The important things that I want you to take from this slide are 2. One of them is that the people we recruited for the study were extremely healthy. If you weren't healthy, according to your blood test, you could not be included in the study. And this is triglycerides, cholesterol, and so on. The other thing is that the people were limited to consumption of fatty fish, marine life, basically before the study and throughout the study, in order to make sure that we can actually look at the absorption of the omega-3 that they received during the study. Now, just reaching the results part of the study, one of the first things we had to do before we even started looking at the groups and comparing them is to look at the baseline values of all the participants and to make sure that the washout worked well. Because if the washout didn't work, we really can't compare them if they had difference in baseline. So we used 3 different tests to look at the groups, the baseline, and we were very happy to find out that 8 weeks of washout was sufficient to actually have everybody at the same baseline. The statistical test was analysis of variance. We first looked at the group because there were 3, like krill oil, fish oil, and placebo. So first we used analysis of variance to see if there is a difference. And then if there was a difference, a significant difference, then we compared fish to krill, fish to corn, and then krill to corn. So this is the first result. Now the results I'm going to show you have, each one will have the plasma and the red blood cells. The blue bars are always the baseline. The red bars are endpoint. And the stars here indicate statistical significance. So from now on, I'll say significance. It means it's statistically significant. And another thing is that throughout the results, we always had the same significance for both the plasma and the red blood cells. So EPA. EPA is absorbed better in both the plasma and the red blood cells by krill compared to fish. Very significant. As you can see, the p-value is extremely low. On the other hand, when you look at DHA, we saw that there is no difference between the krill and the fish, even though there is some significance when you compare the krill with the corn here and here, but nothing between krill and fish. And this is a very interesting result, and I'm going to discuss it later. When we look at the total omega-3, and I remind you again, the total omega-3 was exactly the same given to the fish and the krill. Again, the total omega-3 that we see in the plasma, okay, and in the red blood cells is higher when it's given with krill compared to fish oil, and in a significant way. So this was one of our most interesting results, and this is the omega-3 index. So what is the omega-3 index? This is actually the sum of EPA and DHA in percent out of the total fatty acids in our red blood cells. And why is it so important? I will also discuss later. But we really wanted to know, can really krill oil change the omega-3 index in our bodies better compared to fish oil? And the result, again, is that it does. Krill oil is significantly better than fish in the red blood cells and also in the plasma. This is actually the same result that I showed you before, but this is a different presentation because here we're looking at the change, okay, just the change. And as you can see, krill changes the omega-3 index by 1, while the fish by less than 0.5, suggesting that krill can change your omega-3 index twice as much as fish does in the same amount of time for the same amount of omega-3. So this is just to summarize. Again, I showed you that in both the plasma and the red blood cells, we have better absorption of EPA, of total omega-3, and of EPA plus DHA in the krill compared with the fish. This presentation gives in percent change, okay? And this is significant for all of them. Additional tests that we've done was to try to look at the difference between genders, men and women, and to see if one group has better absorption of omega-3. And we also looked at different age groups, and in both cases we found that there is no difference. We also looked at safety parameters, blood tests, physical examination, and we found that krill oil as well as fish oil are very safe, and there are no serious adverse events reported. So to conclude, this is the first study that tested bioavailability of omega-3 from krill versus fish oil that had identical levels of omega-3 in both treatments and looked at the red blood cells. Second, the objectives of the study were fully met. We demonstrated that supplementation with krill oil significantly improved absorption of omega-3 fatty acids, of EPA, and of EPA plus DHA, which is the omega-3 index. The results were fully consistent. We saw, first of all, similarity between the plasma and the red blood cells, and also when you look at other studies that were published before, a lot of the results were consistent. So just a short discussion with 3 questions that come to mind. First of all, why is the measure of levels of omega-3 in the red blood cells so important? Second, why DHA has not shown the same effect as EPA? And lastly, what is the clinical significance of the change of omega-3 index? So I'll go briefly now because I'm out of time. The plasma levels of omega-3 fatty acids are indication to a short-term absorption. However, the red blood cell fatty acid composition reflects long-term absorption of EPA DHA. Not only that, but absorption into the red blood cell membrane reflects absorption into target organs such as the cardiac membranes and more such as the heart, the liver, and so on. Omega-3 index is determined by percent of EPA plus DHA from total fatty acids in the red blood cells. So this measurement is really very important clinically. Now, why the DHA has not shown similar effects? So if you can look at the graph here, I borrowed it from Cao et al. That were published in 2006. So our finding that EPA is absorbed better is actually not new. As I said, another study that was done recently also showed a trend for EPA and nothing for DHA. In this study, they looked at 8 weeks of absorption and then washout. And this is EPA, this is DHA. And also the authors of this study concluded that EPA is much better absorbed compared to DHA. And one of the reasons suggested is that the level of DHA in the blood is very tightly regulated. So it's more difficult to get it to elevate and change rapidly. Also, if you look at the levels of DHA and EPA, and this is also consistent with my results, you'll see that the level of EPA in the baseline is low compared to DHA. So it is possible that because the red blood cells and the plasma are more saturated with DHA, it's more difficult to show a change. You need longer time or maybe higher quantities. Another thing is that krill oil has higher levels of EPA compared to DHA, so that could be also another reason. Second is the omega-3 index. So the omega-3 index is now a new risk factor for risk of sudden cardiac death and cardiac disease. So this is a graph that was manufactured by Harris, which looked at a great many studies that were done and compared the levels of omega-3 in the blood and the risk of people to get here you can see this side shows for primary cardiac arrest, and this one shows for cardiac death, where here you can see the risk, okay? And here you can see the level of omega-3 and the red blood cells. And what you can see here, that the higher the rate, the percent of omega-3 in our blood, the lower the risk for either having a primary cardiac arrest or dying sudden death from for cardiac reasons. And based on this, he actually produced a scale with so far, at least for now, until further studies are done, the cut number here is 4%. So you want to have the level of EPA plus DHA in your red blood cells to be over 4% in order not to be in this undesirable zone of high risk of cardiac disease or death from cardiac disease. And you really want to push it over the 8 to be in the desirable zone. And one of the interesting things about the omega-3 index that unlike other risk factors which we cannot control, such as age, diabetes, or even cholesterol levels sometimes, this is a risk factor which we have full control over by simply either eating fatty fish or supplementing our diet with omega-3. We can actually push our levels of omega-3 index towards the desirable zone and protect ourselves from those risks. So by this I'm finished. If you have any questions.